Well, good afternoon, guys. Just thought I'd better podcast all these new PowerPoints and replace my old ones. So we obviously started here last year in orientation week. If I can get that slideshow up, that'd be good. There we go. With the plasma membrane. So let's start there, and I'll work through our first 6 podcasts as quickly as I can. So clearly, some of the basic stuff we need to know about, then it may not necessarily be examinable, but it's behind everything you're doing, is this idea of cell theory. All living things are composed of cells. All cells arise from a pre-existing cell, and the cell is the smallest living organisational unit. You should know that. That's good Year 11 stuff. It's stuff you need to keep in mind through a lot of the stuff we're doing. This is all about cells. This first area of study is particularly all about cells, but really everything we do is. And cells are comprised of smaller components, and these smaller components are called organelles. Some of them contain their own DNA, and there's a lot of theory around that many of these things actually have sort of come together in what we call eukaryotic cells, the more advanced cells like our own, from prokaryotic cells and other cell parts that were sort of around at the start of life 4 billion odd years ago. So of course, these things you will have done in Year 11, and I've asked you to revise them over the summer holidays. We have a plasma membrane around the cell. It's a biphospholipid layer. We'll talk about some of the details later. Nucleus contain DNA in eukaryotes, or just free-floating DNA in prokaryotes. The cytosol is the fluid that exists around the organelles, and often called the cytoplasm if we're talking about the organelles and their fluid, excluding nucleus. That's an important little caveat. Mitochondria, endoplasmic reticulum, ribosomes, Golgi apparatus, centrioles, lysosomes, vacuoles of varying sizes, vesicles. These are all found in all plant and animal cells. But we also find plastids, and of course the most famous plastid is a chloroplast, large vacuoles, cell walls, and a thing called a tonoplast, and these are all found within plant cells. They make them a little bit different. The single most important defining feature of a plant cell is the cell wall. Not all plant cells have to do photosynthesis to make glucose, so they lack chloroplasts, and the classic ones, of course, the roots, they don't see any light. So the cell wall is a classic defining feature that you need to be aware of. The earliest cells lacked membranes around their bits, so everything was free floating, particularly of course the DNA. So all the biochemical processes just occurred in the cytosol. This limits the size clearly, and probably limits the ability to evolve and change rapidly as well, and to respond to attack. Our modern cells, like our body's cells, the more highly evolved ones, are membrane bound, so all the organelles are wrapped up with a membrane of their own. So we call those earlier cells prokaryotic, and these more modern cells are known as eukaryotic, and that's a definition you do need to know. Generalized plant cell, generalized animal cell, I won't go into any detail there, no point. There are a number of molecules, chemical molecules, that are clearly vitally important to all living things. So we all need water. Every living organism on the planet contains water, between about 60 and 70% generally, some a bit higher, some a bit lower, but generally between 60 and 70%. And that water allows for osmotic gradients to develop, for diffusion to occur, for other things. So it allows materials to move within an organism and within cells of an organism. We need oxygen. Clearly we use oxygen to break down glucose and release the energy stored within it. Carbon dioxide is important in plants, obviously, when they go around making glucose for us all. Nitrogen's a key component of all of our proteins. The mineral salts, calcium, magnesium, phosphorus and sodium, are all involved in a huge range of movement of materials, structural components. All our lipids and proteins and carbohydrates contain these things, and they allow us to function. And we'll go into some of those in detail as the year progresses. Most of the other nutrients required are found in the soil as mineral salts, and they get taken up by plants, and of course we get them by eating them. These are sort of small mineral things like phosphorus, potassium, calcium, magnesium, iron, sodium, iodine, sulfur, which we need a fair bit of. And then things like boron, manganese, zinc, molybdenum, copper and chlorine, which we do still use, but we use them much less. So trace elements within the food we eat is fine. Some people like selling you products that contain lots of this stuff, but you don't need to be buying that. Organic compounds, so these are things that organisms actually make for themselves, and the four groups, three groups in a sense, are carbohydrates, proteins, nucleic acids and lipids. The course used to look quite a bit at carbohydrates and lipids. They're going to be of passing interest in the new course, and our major concerns are going to be about proteins and nucleic acids. Proteins are clearly highly complex molecules, and you can see a very complex one there developing in the diagram. And they're made up of amino acids, which form polypeptide chains. So we often call them they're a polymer. So they're many amino acids joined together, and that first primary protein structure there is simply a polypeptide chain. It's not yet a functioning protein, but as it moves through the stages of becoming one, then we see it folding and pleating and coiling around itself and creating an active site, and that creates that functional protein. Enzymes, hormones, catalysts, structural components of cells, carrier molecules like hemoglobin, protein channels in membranes, these are all obviously made of proteins, so proteins are intricately connected to our ability to function, and we'll do a whole podcast just on proteins later. I'll quickly go through the organelles. You should be doing this for yourself, and you should already know a lot of this anyway, but obviously the membrane is critically important. This biphospholipid layer, one of my neighbours going off there, of lipids, obviously. So we've got the hydrophobic region within and the hydrophilic regions on the outside. So the heads of these phospholipids are attracted to water or attract water to them, and then the tails that form this hydrophobic region within the cell in here are created by these tails that repel water, which makes it interesting when we talk about how water might move across that membrane. And you see also here we've got there's a little pointer. There's the a transmembrane protein, so a protein channel. We've got glycoprotein here marking this as self, so MHC marker, class 1 marker. Some other proteins integrated into the membrane, which may well be docking stations for enzymes and other proteins, hormones, that sort of thing. And what we can't quite see here, of course, is most importantly, is the idea of some cholesterol locking these two bits together. Because a lot of electrostatics will tell us that these two layers should be trying to get away from each other. So the cholesterol and these integral proteins and things help lock these two together. Hence, this is called a fluid mosaic model. And of course, as we always say, the plasma membrane's involved in making sure what we want gets in, and what we don't want doesn't get in, and vice versa. What we want to get out gets out, and what we don't want to get out gets out. So it is a very important part of the cell. We talk about things being hydrophobic and hydrophilic. So hydrophobic, a molecule is repelled by water. Hydrophilic, one's attracted by water. And these lipids, of course, are hydrophobic and hydrophilic at the same time. We can have a polar molecule, so it has charged ends, or non-polar, so we don't have charged ends. And if you're polar, it's very hard to cross that membrane. So non-polar molecules do a much better job of crossing the membrane. And size is important. Small things can simply cross by squeezing through, whereas a large molecule like a protein simply can't get through. And in fact, even glucose, small as it is, it's only 24 small atoms for that matter, can't cross the membrane around our cells. So they're partially permeable, but that permeability can be quite restrictive. Things obviously move by diffusion, and you will have learned about diffusion already, and they cross the membrane by diffusion. So things move from an area of high concentration to an area of low concentration. So in the diagram here, we have a high concentration here of the green solute, let's call it sugar, and on the other side we have a very low concentration. In fact, there is none. So that would be quite a steep. If we drew whoops, I went too far there, didn't I? If we drew that as a graph, we'd find this was a very steep graph, because there's a high concentration to a zero concentration, and therefore things would move reasonably quickly quite early to start balancing that out. Water, of course, has this special way of moving, and it's a form of diffusion, but it's sort of anti-diffusion, if you like. So water molecules move from an area of low concentration of the dissolved substance to an area of high concentration of the dissolved substance. So in this case here, we have lots of dissolved sugars, and over here we have few dissolved sugars, and so the water will move from this side to that side. Now, if you're thinking about this, that's water moving from high concentration to low concentration, but it only does it because of the concentration of the solute, and so it's doing it in relation to the solute. So it's a little bit different. It's a very important distinction to be aware of. They do like to try and catch you out. We talk about things being isotonic, hypotonic or hypertonic, and here we're seeing an isotonic situation. So the waters, little purple dots, and the solute, the pink dots, are in equal concentration either side of the membrane. Now, that doesn't mean there won't be any movement, but movement's going to be slow, and movement's going to be maintain that balance. We won't see a great deal of change in that. If we're in a hypertonic situation, we're seeing much more water inside this membrane and lots of solute outside. So by osmosis, the osmotic gradient would set up, and this water would move out against the concentration of the solute. If the membrane was available for the solute to cross, the solute of course will also be crossing in high concentration to low concentration and find that isotonic situation again. And hypotonic is the reverse. So we've got very little water inside the cell here, or membrane, whatever it is, a vacuole, and lots outside. Lots of solute in, not so much outside. So the solute's going to move out by diffusion, and the water's going to move in by osmosis. So you just need to be aware of those terms. They may not be examinable, but they're things that are occurring underneath all the stuff we're talking about. How am I going for time there? I'm going to run out, aren't I? We talk about ways of crossing this membrane. So we talk about protein-mediated transport. So obviously osmosis and diffusion don't require any energy. That's happened by themselves. Facilitated diffusion, again, doesn't require energy. So things will just move down a protein channel because a channel exists for them to do that because they're too big otherwise, or they're polar, or they're not lipid soluble, so they need another way of getting across. If you need to spend energy, you can move things across concentration gradients. So you use the idea of active transport to move across the concentration gradient, and the idea of the sodium pump here works that way. So we've got facilitated diffusion. Oops, I just went backwards. There we go. Facilitated diffusion here, and we've got this channel that allows this particular product to come through. No cost, high concentration, low concentration, it'll move in. If the high concentration's on this side and low concentration's on that side, it'll move out. The pump's there for that purpose, or the channel's there for that purpose. The sodium potassium pump is one of these classic active transport pumps, allows for movement against the concentration gradient. So we might have lots of sodium here and not much, and lots of sodium over here, but we want the sodium out, and so we have to move it against the concentration gradient, and it's going to cost us ATP, it's going to cost us energy to do so. And the nerves work that way. We'll talk about nerves a bit later in the semester, and how this sodium potassium pump runs along nerve cells carrying the charge. But we'll talk about it later. I have actually loaded this for you, I hope. Maybe I haven't done this one. I will make sure this is loaded on the sector as well, because there's really nice little animations that I've attached here. I think McGraw-Hill or something, where they originated those, but they're really good little captions explaining all four of those things. And that is our first podcast for the year. So I shall stop that.